Thank you for coming today. My name is Thor Rollins. Some of you may have seen that Audrey Turley's name was on this presentation outside. Audrey originally planned to come and present. She had a family emergency, so you guys are stuck with me. But hopefully, we can still do this pretty well. So today, our presentation's on the new ISO 18562 standard that is coming out. It just recently came out. This standard is actually quite interesting. It's one of the first big impacts in the gas path industry that we've seen. To give you a little bit of background, so I've been doing this for 15 years. When I first started with gas path devices, almost no testing was done on a gas path device. In fact, it was kind of considered as a non-patient contacting device. If it was tested, it was just minimally tested at all. About 3 or 4 years ago, we started seeing the FDA require a bunch of testing for these types of gas path devices. So they started classifying them as external communicating devices with permanent contact, and they wanted to see even implant, which didn't make sense, and some other things. And so we started having a developed cycle with that. About 4 years ago during this show, we met as a TC194 group, which is the ISO committee group for biocompatibility. We met with the ISO group over gas paths, and we actually helped write the 18562 because the FDA was having all these concerns regarding the biocompatibility of gas path devices. So this standard has now just become enforced, and we're going to talk a little bit about today of what we're seeing from regulatory agencies on what they're wanting to see based on how your gas path is. Also, some things are going to change because like any new standard, there's some flaws in 18562. So we're trying to get around those flaws too. Am I doing this wrong? There we go. Does that work? It's counterproductive. Okay, thank you. Okay. So first off, before I wanted to, because this is going to play a role in today's presentation with the chemistry, just recently Nelson Labs purchased Toxicon Europe. Toxicon Europe is a very advanced chemical characterization company in Belgium. They have been focused a lot on pharmaceutical industry, container closures, but this type of work plays heavily into the new standard and the impact that we're seeing from the regulatory authorities for this new standard. So we just purchased this company to help not just with gas path devices, but with the chemical characterization of all devices. So that's recently. They do have, and it's going to be our dedicated European Center of Excellence. So we're going to have a place for all our European customers to be able to ship samples instead of shipping it overseas to us. So that's some good news for us. Okay, so these are the standards that we're going to be talking about today. Of course, we're going to be talking about 18562 and how that impacts. But remember, 18562 is very focused on specific types of biocompatibility. These devices still have to look at the suite of ISO 10993 and to do that type of overall biocompatibility assessment. And the new US FDA guidance that came out in 2016 plays a huge role in this type too. They have guidance in there on how they like to see gas path devices. But just like all biocompatibility, this new gas path standard relies on a risk-based approach. And it's something that we have to kind of take into account when we're evaluating the biocompatibility, just not of gas path devices, but of every medical device. This is what the new standard, which if you're doing biocomp, you should be familiar that the new ISO 10993-1 is in public draft right now. In fact, you can get that. There's a link, and if you need that link, you can let me know. Oh, by the way, I want to let you guys know, if you want a copy of this slide deck, I'd be happy to give it to you. If you just give me your card afterwards, I'll email you tonight a copy of the slide deck, and I'll send you the slide for the public ISO draft 2. So ISO 10993-1 is in public draft, so it's a brand new standard. This new standard is relying more on a risk-based approach for biocompatibility. So this type of work is what is needed going forward for all devices, which is you need to do an initial risk assessment, including a biological evaluation plan. So that risk assessment talks about what's the risk of your device and how you're going to mitigate those risks by either testing or some kind of assessment. So as soon as you have your initial BEP or plan, then you move to whatever testing is in that standard, or in that plan, excuse me. So if you're going to do chemistry, or if you're going to do the historical biocompatibility test, whatever is in your plan, then you actuate those plans. And then you have to develop a biological evaluation report at the end. That's a BER, and that's in the FDA guidance document. So basically, the whole setup is you have your initial risk that helps you develop a plan, you act on that plan, and then you sum up your findings in a report. So this is the way we need to evaluate our biocompatibility, and it makes sense, but a lot of people were just checkboxing for a long time the biocomp tests. And now we have to do a risk-based approach, which means that we're actually able to justify out of a lot of testing that historically we weren't able to justify when the FDA required a checkbox approach. And we're actually getting better and better response from the FDA on this. The more and more we do it, the more and more the reviewers see it, and the more comfortable they are with some of the justifications that we make. So, with this 18562, though, that's the same concept we need to apply, which is this risk-based approach. We need to see how this gas path device is going to have a risk to the patient. We need to set up testing that we feel is appropriate based off this standard and the ISO 10993, and then look at the results to be able to talk about the safety of the device. So the whole thing is about the risk to the patient versus the benefit to the patient. So this is the risks portion of the new FDA guidance document. If you haven't read this, I would recommend at least going through it. Don't do it too late at night because it's not one that keeps you awake, but it's still like most standards. It gets good information. I actually really like this guidance document. The guidance document that was before this one was a 1995 document. So as you can see, the amount of time between the initial guidance document that we were working on, the G95 document, to this one is a huge difference. I mean, 1995, I was graduating high school, so that was a long time ago. So this document is actually a really good insight of what the FDA's current thinking is on biocompatibility. It's not perfect, like any government document, but it's a good document. It's much better than the document we had before. So please be aware of this one, even when you're looking at the biocompatibility of gas path devices. Anytime we deal with medical devices, like I talked about, we have to evaluate the risk of that medical device. The new ISO 10993-1 is going to deal a lot with the 14971, okay? So when you're looking at risk, this is the document we need to have in our back pocket to be able to understand the risks of this device. I have customers that actually have their internal protocols where they sit down and look at biocompatibility. They say, what's the probability of occurrence of harm and what is the severity of that harm? And that give them that number to help them decide what kind of testing they need to derive based off that risk assessment, okay? So there are ways to evaluate risk using this 14971. And the new ISO 10993-1 actually relies heavily on the standard. So it is something we should have in our toolbox. But I also want to talk about with this risk-based approach, it's not new. It may be new to a lot of customers who've been checkboxing biocompatibility for the last 4, 5, 10 years, but the approach isn't new in the standard. So this current flow chart has been in this ISO 10993-1 standard for a while. And it really is your initial risk-based approach. It goes through questions to help you understand the risk of your device, like, is there a device on the market that has similar materials and processes, that have similar contact types? So you're able to go through this kind of initial risk-based approach to be able to understand the risk of your device and understand what testing is needed to mitigate that risk. This is that first approach that we talked about, the first steps, but it's been around forever. It's just the new -1 is trying to emphasize that type of approach. So this right here is the biocompatibility chart from the FDA guidance document. Okay, so the one that just came out in June. There are some differences. They've added material-mediated pyrogen. Even though they've always requested material-mediated pyrogen, they've added it as a category. But then one of the biggest changes is how we look at those circles. Now, the circles have always been there, but in the past, the FDA didn't always enforce those circles. They may if they had concerns about your device, but it wasn't a constant enforcement of those tests. Now, since we're not checkboxing, right, we now have to evaluate the risks of all those endpoints. Those circles are no longer optional. Oops, I thought I had another one there. Are optional. The FDA says those circles are endpoints that are required to be considered by the FDA. The X's are endpoints that need to be considered per ISO 10993. So basically, all the circles and X's are the same, which means you need to consider why or why not those test endpoints are appropriate. Now notice, I didn't say you had to test those endpoints. In fact, please don't checkbox these tests. What you need to do is that risk assessment and then see if these endpoints are valid for your device based off that risk assessment. Okay, so your plan needs to tell you what of these tests are actually appropriate and which ones are not and why not. Okay? This same approach, though, is also intended for the gas path standard. All medical devices need to have this risk-based approach. So the difference between the US FDA and the ISO 18562, though, is really the difference between the gas path and how we look at how it contacts the device or the body. So with 18562, the influence with the gas path is the indirect contact with the gases and particulates into the lungs. Okay? The FDA may have some additional concerns based on how the liquid may be condensed or how the device may be used. And we'll get into those in here in a little bit. So this is the basic setup of 18562. There's 4 parts in the standard. Okay? The first part has to do with the initial risk evaluation, kind of like we talked about already, an important part of all medical device toxicity testing. The second part has to do with particulates. Now, if you're thinking about the risk of a medical device, when you're thinking about gas path, particulates are a legitimate risk. When you're breathing in gases, if you're shedding particulates from a plastic, from an adhesive, or something like that that are being lodged into the lungs, that causes a legitimate biocompatibility concern. So we need to evaluate particulates for gas path devices. Then, depending on the risk, now remember, these sections 2, 3, and 4 are all dependent on your initial risk assessment. So we're not necessarily have to run each one of these sections only if your initial risk assessment indicates that these are risks, okay? So section 3 has to do with volatile organic compounds. These are things that evaporate, compounds, chemicals that evaporate off very quickly, which also makes sense for a gas bath device because you're breathing in the air. So the air can take these chemicals and they can leach them off of these plastics or glues or even residuals on metals and deliver those into the lungs of the patient. So VOCs are important to evaluate when you're looking at toxicity. Remember, gas is just like it's a liquid carrier. It acts just like a liquid carrier. So just like a device that might sit in the blood can extract chemicals off to go IV, gases can extract chemicals off of plastics and deliver them to the lungs just the same way. So that's what we're evaluating. Dash 4 has to do if you have the possibility of condensate in that gas path. So if you're using humidified air and there could be some kind of buildup of that humidity where now the condensate or liquid, touches the patient or goes into the mouth of the patient, we need to evaluate that risk. Okay? So these are the 4 parts of the 18562 standard. And each one, this part 2, 3, and 4, is ran if the part 1 initial risk assessment is deemed those parts are necessary. So we're going to go through a little bit in detail. This is more for your information. Like I said, if you want a copy of this deck card or deck, give me your card. But this is our flow chart on how we walk through 18562. Based on the initial risk assessment, what we asked ourselves, what questions, what testing we would then go through based off the answer to those questions. Okay, so it's a good tool, I think, going forward for people who have gas path devices. But then, once we start asking these questions, we put them into our biological evaluation plan. So this is a format of what we include into when we do a biological evaluation plan. But that initial gas path questionnaire needs to be put into here to be able to understand what's your design for your biocompatibility for that device. The next step is the actual testing, right? So we've already evaluated what risks and what testing we're going to do to mitigate those risks. Now we're actually going to talk about what tests are required. So when we go back to looking at our section 2, 3, and 4, these are the tests that are in those sections that if we deem they're necessary, we have to run. So for dry air pathway, okay, we use section 2 and 3. 2 is particulates in the air, and 3 are those volatile organic compounds. Those are what's required for a dry gas path device, along with your other biocompatibility endpoints, right? Okay. Now your humidified air path, though, if you have humidity in that air, we have to do a little bit more. We still have to be particulates. We still have to look at VOCs. SVOCs in air may be required because, remember, water, if it's humidified, may be drawing out more than just the volatiles. It could be drawing off some semi-volatile compounds. Also, we now have to look at the condensate issues. Is that water drawing off different compounds in the air? So now we have to do some more traditional extractable leachable tests in water to be able to determine the true impact of the safety of that medical device. I do also want to point out, and we've been very successful with this, there in the bottom, with this type of air gas path, we're able to evaluate sensitization and irritation using a structure analysis of those compounds. So basically the way I like to explain it is it's like going to a lineup for some kind of a robber or some kind of a bad guy. You're sitting there looking at a lineup and you're picking out which ones you think are guilty, right? That's what this QSAR method is we look at all the compounds that we saw come off your gas path and we look at how they look, okay? We look at their structure and we say, yeah, that one looks like it's a bad guy. So we're gonna say that one could be a sensitizer based on how it looks, or that one could be an irritant based on how it looks. So we're able to evaluate the sensitization and irritation potential of the humidified gas path without running animal tests, okay? And this makes a big difference because remember, with a regular medical device, you're putting in contact with skin or with blood or with tissue, and that becomes a little bit harder. But with gas path, you're looking at just what's pulled off with the gases. That's all you're concerned about. And so we're able to evaluate that using this chemistry more effectively than we can with other medical devices. Yes? So if your gas path device does not contact the skin in any other way, then yes, we would be able to use this instead of doing the sensitization and irritation tests. If you have a mask that contacts the skin, then that part would have to go through the traditional biocompatibility tests. And just so you know, so only if no surface contact, that was a segue, good job. So this has actually been very successful for us. We've done this with the FDA in the last 4 or 5 months and have been able to be successfully argue that this is a good approach for these types of devices. So we still need to be able to do this plan, though. We still need to know a lot about the materials and processes that go into your medical device. This is part of that initial risk assessment. We can't really do a good job of deciding what testing and how it's needed unless we know what's there, right? And this is becoming a much bigger responsibility for medical device manufacturers, is understanding the materials that you're putting in your device. Before, if you just passed a test, you felt good about it. But now we have to be able to evaluate these tests appropriate, what tests are appropriate, and we can only do that if we really know what we have to begin with. So please really get good relationships with your material suppliers. If you're a material supplier, be prepared to give over more information to your medical device manufacturers because we're starting to see this become more importantly when you do a risk assessment to be able to have that relationship with the supplier of your material. So the first step is this particulates. How do we test for particulates? Now, there is a test that a lot of people are familiar with if you're in the vascular space, where we run water, particulate-free water, through the device and look at how much particulates come out. We can still kind of do that if you want, but it's not clinically relevant if you're doing a dry gas path device. So the results may not mean as much to us. So we actually have a different method that we can look at particulates by weighing them out with a very sensitive scale on a filter. So basically we run your device like it normally be clinically ran. We collect the particulates that are lodged on the filter, and then we weigh that filter difference. And this helps us evaluate about the particulates that have been generated from your device. But then there is no good acceptance criteria for particulates. If you're looking for one for a medical device, the best one we have is this USP acceptance criteria. This is the results. We use this one, but this is for containers for injectables, and it's just not great. But the FDA has allowed us to use these limits to help to prove safety. Okay? That doesn't mean that if you're above these limits that it's automatically unsafe. It just means if you're below these limits, we feel good about justifying the results. If you're above the limits, then we may have to do a little bit more of an evaluation on the results and what it really means. Okay? It's one of the biggest holes in this testing is the particulates because there's just not good limits for medical devices out there. The next one we have to look at is these VOCs, these volatile organic compounds. There's actually a kind of different ways to be able to evaluate this with gas path. Remember, this is dry gas path to begin with. The way we like to do it, and we find it's best, is we actually use these canisters. So we run the gas through the device like it's clinically supposed to be used, and we collect the gas in these canisters. So all the chemicals that are leaching from the plastic are being held into these canisters. Then we can take these canisters, put them onto a GCMS, and be able to read what's in there. Basically, try to identify and quantify the compounds that are leaching off that are stuck into these canisters. This is much better than running fluid through it to be able to catch the fluid to look at what volatiles or heating up the whole device, right? If we put that whole device under a glass case and heat it up, well, that's not clinically relevant because now all those chemicals from all that material is leaching into that test. And that's, if you pass, that's great, but if you fail, what does that mean, right? This way, we're able to get just the gas path tested under clinical conditions. So once we get we use the STM method to do this. That's the canister method. Once we're able to get the canister in there, we're able to identify and quantify the compounds that we see that are coming off the gas path. But remember, you need then to have a toxicologist evaluate those results. As much as tempting as it is just to give the FDA a sheet of chemicals and say, look, we only have 4 of them, so it's got to be safe. We've had customers do that, okay? The FDA is going to say, please have a toxicologist evaluate this. Luckily, we have a lot of data about inhalation studies. It's one of the most studied types of compounds is inhalation. So usually we can find inhalation studies for these types of devices. As a toxicologist, you're able to identify it, look at the dose, because remember, dose makes the poison. Everything's toxic if it's high enough, right? So we're able to look at the dose and then determine if it's an appropriate safety margin or not, okay? And if you guys want to shut me up, the best way to do it is just to raise your hand and ask a question, because that makes me stop talking, as you can tell. If not, I just keep on going. So that part there, that was for dry gas path. Now, once we add humidity, humidified air, we open up a whole different realm of testing. Now we have to worry about just not the gas leaching chemicals off or particulates off. Now we have to worry about the humidified or even condensate doing a different job. So that moves into the other section. Now, this is where we think we're going to change the standard. Once again, once you start writing a new standard, you think it's good until people start using it, and then you start realizing where the gaps are. And this is one that I feel like is a gap. I don't feel like it's written well right now. So when we revisit this standard, this is something that we probably will change. But we still want to test for leachables under this type of use, right? This humidified air. What we recommend is a worst case approach. Now, we still don't recommend, so we recommend an extractable leachable test per ISO 10993-18. We don't recommend that you cut up your whole device and put it in there, because once again, that's not clinically relevant. We recommend running water through the gas or the humidified gas path to look at under the clinical conditions to look at what comes off. Now, that's still worst case because you're using water instead of humidified gas. But it's most of the time we end up having appropriate safety margins and we're able to justify it as worst case. Still would not, though, chop up the whole device and put it into a container and extract it, unless that's how your device is used, which I wouldn't think so. So this is how we would recommend it. So this is our typical extraction. So once again, this is just an example of a -18. I would run the water through the gas path. But this one, we extract the whole device into the water. I don't know if you can see that, but that's Leonard Nimoy's Star Trek hand, okay? So we extract it in water. We recommend a clinical aggressive extraction. We do have up here 50 degrees C for 72 hours. That's worst case. A lot of times we don't use that because, let's say it's done at 37 degrees or at room temperature. Most of the time if it's humidified, it is at 37 degrees. But we want to mimic worst case clinical conditions. So then we run it through our VOC, SVOCs, maybe non-volatiles, but mostly what we look for is volatiles and semi-volatiles because that's what we're probably going to see from the humidified gas. Then we get our chromatogram, and that basically just tells us what the types, depending on how good your library is, it tells you what's coming off and how much of it's there. And then you're able to identify and quantify to be able to do, once again, a toxicological assessment. You need to do a tox assessment on this too, because the information doesn't help you unless you know if it's going to hurt someone when they breathe this in. So these are the results that you see. You see a component. We're able, in most cases, to tell you where those components are coming from. Is it coming from a plastic? If it's coming from an adhesive, is it coming from a metal component? We're able to identify and quantify the amount coming off if it's in the library, which more and more compounds are becoming a part of the library. And then we do the toxicological risk assessment. So just to give you a very high level of how we do a tox assessment for these gas path devices, we do it per ISO 10993-17. If you ever wanted to play at toxicology, it's a great standard to read if you can think about how we do it. But basically what we try to do is we try to, for each compound that we find, we try to find a study done on that compound through an inhalation route. So we try to see if some reputable organization has done a tox study on that compound through an inhalation chamber. Then we try to find what's called a NOEL or LOEL, which stands for no observed adverse effect level or lowest observed adverse effect level. Basically, we're just trying to find what level caused no toxicity in that animal. Okay? So they give them different doses and they find where the dose ends with no toxicity. Now we have some kind of idea about what the toxic potential of that compound is. And we don't have to rerun animal tests because someone 20 years ago ran an animal test that we're levying. Okay, so we're trying to reduce the amount of animal tests we need to run. Once we have that information, all we do is math. Okay, there's a little bit of figuring in there too, and I'll explain. But basically, we take that NOEL that we found. Okay, then we do some uncertainty factors. Basically, the first uncertainty factor is that people are different. Okay, like some people are wimpier than others, right? And so just because some animal had a reaction doesn't mean that some other animal wouldn't. So we do a safety factor of 10 there. That's standard. We always do a 10 for the first one. The second one is if we found animal data. So if we used animal data and we want to take it to humans, we do another safety factor of 10. It's very rare that we can find people volunteering to have this type of work done on humans. So usually it's animal data that we have. Okay, so right away we have 100 safety factor built into this study. The next one is how good a data do we have? Was it a good study? Was it done inhalation? Was it done on multiple animals? If it's a good study, we use 1. If it's not, we start to use 2, 3, 4, or 10, depending on what kind of quality of study we have. So that's the only part of this that you may need a toxicologist input on. But as soon as you have that, then you're able to come up with something called a TI, which is a tolerable intake. Okay? So that's how much that we can take in based off of those safety factors per day. That's breathing. Then we take our TE, which is just our TI times the inhalation rate, and then we do some uncertainty factors, but basically we try to get a margin of safety. So we're looking at how much can we breathe in of this compound and be safe. And that's that we do that for every single compound. Okay? That's the conclusion we try to get to, which is everything is safe, right? So there's some possible considerations. We want to talk a little bit about the surface device. If you have skin contact, what does that mean? we still have to run our if we have skin contact, we still have to run our traditional biocompatibility tests. And usually that means that you have mucosal or skin contact, so you're looking at the big three, right? Cytotoxicity, sensitization, and irritation. Making sure I have time. So the cytotox test, I'm just going to run over these really quick. Actually, the cytotox test is probably the one you should know most about, because the cytotox test is your best friend and your worst enemy at the same time. It's your best friend because it's the cheapest and the quickest test, but it's also the most sensitive. In fact, at Nelson Labs, when we look back at failures at Nelson Labs, 93% of the time, if you fail a biocompatibility test, is this test. Okay? What that means is that if you run this long enough, you're going to fail this test. But that doesn't mean that's bad, okay? Cytotox fails. Silver, copper, zinc, all those fail. Citrate plasticizers fail, but they're not necessarily unsafe. But it's a good screening test. So if you're able to pass this test, you have like a 90% assurance that you're going to pass the rest of the tests. Okay? Good screening test, but not necessarily if you fail it, doesn't mean your device is unsafe, okay? So this is how we evaluate the cells. We put the extract of your device on the cells. This is a 0, which means it's all healthy and happy. They've all grown and nothing's wrong. This is a 4, okay? This means this is latex. Latex is our positive control. As you can see, it pretty much decimated the cells, right? There might be some things left behind, but that's actually the skeleton, because latex is a fixative, which means it burns the cell to the bottom of the plate. So that's not good, okay? So that's the difference between a 0 and a 4, between a pass and a fail. These are the scoring criteria for cytotoxicity. 2 or less is considered passing. 3 and 4 is failing. The next test we have to look for is irritation, okay? Irritation is an immediate response to an antigen. And that's why we have to worry about it. And the best example I have for this one is a real life example. And I'm a little embarrassed to talk about it, but I do it always to help people remember. So my wife wanted to go away with some friends. She left me at home with my 3 girls. I went to the store to buy some detergent. I found the cheapest detergent I can because I'm a guy. I bought that detergent, came home and did some laundry. I washed pajamas. I put my kids in their freshly washed pajamas, and I put them to bed. The next day when they woke up, they had red spots all over their body, okay? That was an irritation response. Not the same irritation response that my wife gave me when she got home, but it's still an irritation response. So this one has to do with all of us. All of us have had irritation responses, right? So this is an immediate reaction to a stimuli. And we do this one in rabbits, and we observe them after we expose the device to the rabbits. We just look for 24, 48, and 72 hours, and we look for that redness and swelling. So that's an irritation test. The sensitization is more complicated. It's type 4 hypersensitivity or delayed hypersensitivity. The best way to explain it is poison ivy. Everybody familiar with poison ivy? If you haven't, go hiking, and you may be familiar with it later. But basically, poison ivy is a sensitizer. You still have the same response, the same redness and swelling as you do with an irritation. The difference is how you get it. So with an irritation, you never had to be exposed before. The first time you contact the irritant, you break out, like my kids with the detergent. But with a sensitizer, it's repeated exposure. So the first time you touch poison ivy, you don't break out. It's the next time or the third time you contact it, because you have cells that remember that sensitizer that are staying around waiting for you to be sensitized. So this one is also ran, but it's a different, even though it's the same redness and swelling, it's different because of how it's performed. These are 2 versions of the sensitization test. The only thing I'm going to say about them is that the LLNA is starting to become out. We're starting to not run LLNA because the FDA is not a big fan of it for most devices. So the guinea pig maximization is the most common one ran. We are starting to be able to use chemistry to justify sensitization, but we're not quite there 100%. I have one big customer of mine that every time they submit to the FDA, they submit using chemistry for sensitization and not animal tests. And then about 50% of the time, the FDA accepts that. The other 50%, the FDA asks them to do maximization tests. So we're slowly starting to convince the FDA through literature, through studies, that the chemistry can predict sensitizers just as good as a guinea pig, if not better, than a guinea pig maximization test. So the last thing before I close up is this endpoint, which is coming up with a report. So this is our version of the report that we do if you contract through us. It basically walks through the initial risk assessment, what testing we did, what the results were, and then the conclusion is why we feel like this device is safe. So when you submit to the FDA, you have your initial plan, you have your test reports, and your BER, and that's your dossier that you submit to the FDA. Works really well. Okay, so that's my presentation. I do want to show you guys, this is the brand new ISO chart that's in the draft, the public draft, but it's in cool slide rule because engineers like to move things. So if you guys want one of these, they're down here. So you can pick one up. And then if you do want to copy the presentation, like I said, bring me your card and I'll email it to you. Any final questions? Yes?
So for extraction testing, how do you decide if your solvent is water or something else like that?
Yeah, very good question. So with a typical extractable leachable test, we would use both a polar, a mid-polar, and a non-polar to do an extraction conditions. But for humidified air, if it's just humidified with water, which most of the time it is, if there's no drugs present in that, which sometimes that's where we have to change it. But if it's just humidified with water, then we would just use water as our solvent. But if there's drugs added, then we'd have to look at how the drug is to be able to determine what other solvents we'd have to use.
You mentioned that's new. Is that new compared to last year?
Good question. I don't remember. So this one's new because there's a new category per ISO 1093-1, which is material characterization. I don't remember if we had these ones yet last year or not. But the FDA, the new ISO 1093, is going to require you to do material characterization, and everything else is going to be E, which is evaluate. So that's the new slide rule. That would be the only thing to be different if it's different from last year. Any final questions? Okay, well, thank you for your time, and thanks for having me.